Hello, everyone. This is Kelly Eversole, the Executive Director of the International Alliance for Phytobiomes Research, and this is our webinar series. Today, we're very honored to have three distinguished scientists from INRAE in France who will be talking with us about Holobionts. And the first person who you'll hear from is Emmanuelle Maguin, and then Emmanuelle Blouin, and then Claire Rogel-Gaillard, before we get into the webinar, I'm going to give a brief overview of the Phytobiomes Alliance and then turn it over to Emmanuelle. So thank you for joining us today, and we hope you enjoy this webinar, and I'll see you again at the end as well. So, all right. So the Phytobiomes Alliance is an international alliance, a nonprofit consortium of industry, academic, and governmental scientists. We have a number of sponsors, including INRAE, and they make it possible for us to actually have this webinar series. I know today our focus is going to be on Holobionts, but I would like to just remind everyone what we mean by phytobiome. And it is a complex system of plant-based agriculture. It's a little different than the holobiont, but the holobiont is certainly a part of it. But it's the concept of a plant in a specific environment and all of the geophysical and biological components that interact with that plant. And then even most importantly, the phytobiome includes the management practices that influence everything going on in that particular biome. The vision is that for us is that by 2050, all farmers, regardless of size, will have the ability to use predictive and prescriptive analytics based on geophysical and biological conditions and use that information to determine the best combination of crops, management practices, and inputs for a specific field in any given year. We are having our goals as far as the Alliance is trying to bring together these multidisciplinary disciplines and really bring in the breadth of both the physical and the biological sciences, and as well as engineering. And so we will be having a conference in September of 2021, and you can find out more information on our website. So our next webinar will be in January, and it will be focused on predictive biology, understanding and reversing the evolution of antibiotic resistance. And you can sign up for, go ahead and register for that webinar, and actually get more information about all of our webinars by going to our website, and then also looking at our YouTube channel. So just to give you some background on how this is going to work, the webinar is being recorded and will be posted on the Alliance YouTube channel in just a few days. The presentation section will be followed by a Q&A period. If you have any questions, be sure and use the Q&A panel. You can submit your questions throughout the entire presentation, and then we'll get to them at the very end. If you'd like to talk to the panel or to your fellow attendees or to the organizers, you can do that through the chat panel. The presentations, I think most of them, or at least two of them, are already on our handouts section of the webinar, so you can actually download those and follow along. The others will be made available subsequently. So with that, I want to turn it over to Emmanuelle Maguin, who will take over from here and give us all of the information about what is a holobiont and how it affects both plant and animal, as well as environmental systems. So thank you very much for joining us, Emmanuelle. I appreciate your taking charge and bringing us up to speed on this very important field.
Do you see my screen?
Yes, I probably need.
Okay, thank you for this nice introduction, Kelly. And in fact, I'm not going to tell you so much about Holobionts on this presentation, but more about a program, what we call a meta program, which has been launched by INRAE on Holobionts and microbial flux in agri-food system. And you will understand that it's a framework in which the two research projects which are going to be presented are developed. So briefly, INRAE is the French National Research Institute for Agriculture, Food and Environment. And we are tackling those scientific challenges, taking into account the context of transition that we have right now, ecological transition, need for more sustainable production of healthy food for all, complementary uses of bioresource in the context of bioeconomy, biodiversity, and adaptive resource management. INRAE is has about 8,300 permanent staff, and our salaries are including in INRAE budget. We are all civil servants, and there is about 3,000 contract staff who are working on contracts. Well, 10 years ago, INRAE started to implement what we call meta program, which are in fact scientific programming tools designed to boost systemic and interdisciplinary research on what we identified as major scientific issues and societal concerns in line with our strategic agenda. The objective is to develop state-of-the-art interdisciplinary research to generate and to consolidate scientific community on these issues, to increase the impact of INRAE at the international level, and to enable new international collaboration on given defined strategic issues. So there were a first period with 8 meta programs from 2010 to 2018 that were at the end positively evaluated by international scientific experts. And since 2019, there is new topics which have been, or new issues which have been defined and programmed. And 10 meta programs will be gradually introduced. So today we are going to show you what we're doing in the framework of Holoflux, which is a meta program that have been launched mid 2019. What can we do in those meta programs? There is some means and incentives. So the meta program is supporting 2 types of projects, what we call exploratory projects, which can be risky projects, but relatively short term, it's 2 years, and the budget is not so high, and bigger project, flagship project, which are supported usually for 4 years. And we expect from those projects to have both scientific and societal impacts. We also support scientific networks to create and to support cross-disciplinary communities. And there is other activities such as workshop, training session, and few PhD students are funded every year. This meta program, Holoflux, is focused on, consider that microbiome are enablers to transform food system toward more sustainable and healthier system through microbiome services such as reduction of greenhouse gas emission, maintaining biodiversity, reduction of fertilizer, and so on. In this meta program, we also consider the paradigm shift associated with the holobiont concept. Holobionts are a host, which is a plant, an animal, a human, or others, and its associated microbial community. And in fact, this concept changed our vision of what is a phenotype. because then you have to consider that individual phenotypes result in fact from the combined expression of the host and associated microbes genomes. So the objective of this meta program, which is launched for 8 years, are to understand, predict, and control the functioning of holobionts and microbiomes, as well as their interaction with the environment in various agronomic contexts, taking into account the issues of sustainable food and health preservation. And we identified 3 main scientific areas on which we want to increase our research capabilities. So I'm going to present them now, one after the other. So one of these areas concerns the mechanisms underlying the assembly and interaction within holobionts. The question we have in mind are to understand the determinants of holobiont assembly, meaning how microbiomes and hosts are assembled, to better understand and to use host microbe interaction mechanisms, to understand microbiome stability within the host, so the question of the evolution and transgenerational transmission, and the impact of the microbiome and microbiome host symbiosis on the phenotypes. For instance, health, performance, immunity, those kind of things. On this area, we already support 6 exploratory projects which have been launched this year for 2 years. And as you see, they are addressing different aspects or sub-questions of these areas. And today you will have a presentation by my colleague Manuel Blouin of this project house, which concern microbial communities in the Rhizosphere. Should we consider it as a selection unit? And a presentation from my colleague Claire Rogel-Gaillard on the enterotype of the intestinal microbiota in pigs, characterization and influence of the host genetic to assemble the holobionts. The second scientific area that we are considering are the microbial flux or flows and dynamics within an agri-food system. And the questions concern microbiota establishment in agri-food system, dynamics and evolution through the system, the impact of different production system, practice or change of practices, for example, decrease of chemical inputs on the agri-food system and on the microbiomes and holobionts, and to identify the critical steps for the sustainability of the agri-food system. And with the identification of those steps, also develop some biomarkers for quick evaluation. On this aspect, we are supporting 2 projects, one flagship project for 4 years, which concern transfers in dairy systems. So the microbial flux at the heart of the agroecological transition of dairy systems, to evaluate their impact on the functioning of the dairy system using an interdisciplinary approach, and an exploratory project which is called Egg-to-Meat, which concerns the transfer in the entire production chain for broilers and comparison of different rearing practices. Then the third area concerns the control and management of microbial fluxes and how they impact the performance, sustainability, and health. And again, we have 3 sub-questions: the levers to modify microbial ecosystem and assess the impact on the whole system. the possibility to sustainably modify the host phenotype by managing its microbiome, and evaluation of the cost-benefit impact of microbial fluxes and management approaches. So in this meta program, the covered area, we consider that the covered area are scientific forefront domain regarding animal and plant production, agri-food systems, ecological engineering, human health, predictive biology, and cost-benefit assessment. It's interdisciplinary. We have mathematicians, microbiologists, microbial ecologists, geneticians, people doing genomics, animal, plant, human physiology, humanities, and social science. And I'm presenting you this, this program here because, as I said, we also want to use it to develop our international visibility, I would say. So we will have, a first meeting of a scientific advisory board composed of international experts next year. There is a possibility to join funded projects or to develop new ones, but I have to say that INRAE will support mostly INRAE teams. There is possibility to create and to support international network with key partners in those areas, and to elaborate joint training or summer camps. And I leave it open to the creativity of all the people. If you want to imagine other aspects, we will of course consider suggestions, proposals. Okay, and here you have the contact for this meta program. So don't hesitate if you're interested or if you want to interact with us. Thank you for your attention. Okay, so as I think the first speaker will be my my colleague Manuel Blouin. Manuel is a professor of ecology at Agroecology Dijon, and is working in a joint mixed laboratory between that engineer school, university, and INRAE. The laboratory is called Agroecology, and he's going to tell us about one of the house program which concern microbial community in the Rhizosphere. Manuel.
Yes, thank you very much, Emmanuelle, for the presentation.
You're welcome. I don't hear you so well, very well.
You cannot hear me? Oh. Can you hear me now?
Yes.
Yes? I will try to speak loud.
Okay. We can see your screen, but you're... Yes.
Okay. Is it full screen now?
Yeah. Yes, it's full screen. That's perfect.
Okay, thank you. Thank you very much. So I would like to thank the Phytobiomes Alliance for giving me the opportunity to present my work, and the INRAE Holoflux meta program to fund this research project, which is called HOUSE. So as Emmanuelle introduced, the title of my presentation will be about the microbial communities in the Rhizosphere, and I will try to answer the question, is the microbial community a selection unit? So you have seen this picture on the presentation on the slide of Emmanuelle, and I want to come back to the seminal paper about the holobiont research, which has been written by Zilber, Rosenberg, and Rosenberg. And so if you know this paper, you know that the holobiont is defined as the animal or plant with all of its associated microorganisms. And the main point of the paper is that the holobiont is presented as the unit of selection in evolution. It is not the plant alone, it is not the animal alone, it's always a plant with its microorganism or an animal with its microorganism. So there is a big debate in the literature on this point. Is an holobiont a unit of selection? In my opinion, it is very ambitious to demonstrate that holobiont is a unit of selection. So I will start by trying to show that microbial community can be a unit of selection because an holobiont is made of a big organism with a long life cycle and many species of microbes with very short life cycle. So we can wonder if it can be a unit of selection. So I want to start with only the small short life cycle microbes and try to see if it can be a unit of selection. So as you maybe know, Levantine has proposed 3 criteria to distinguish a unit of selection. The first one, you need to have variations amongst the entities that you want to select. And the second one, these variations have to be associated with changes in fitness. And the third one, these changes in fitness have to be heritable. So heritability is relatively easy to understand at the organism level. This is in fact the similarity that you can find between a parent and its offspring. But it is more difficult to define at the community level. And one suggestion is that we can look for similarities between parental communities and offspring communities by looking at the stability of the microbial community structure. So the stability is linked with the heritability in this domain of microbial community selection. So now this is a theory how to demonstrate in an empirical way that communities can be selected. So if we go back to Darwin, you probably know the finches of the Galapagos, and this was one of the most famous examples that Darwin gave to say that evolution occurs through selection and through natural selection. Another example in Darwin's books is less known. It's about the pigeon of Darwin. And indeed, Darwin was exchanging and discussing with breeders of pigeon. And thanks to these breeders, he had morphological differences appearing between the different pigeon lines, and also the genealogy, because the breeders are noting everything, and so he can look at the superposition of the difference in morphology and the genealogy, and like this, he proposed the idea of descent with modification. So artificial selection is the main point in the Darwinian theory. So we will use artificial selection to try to demonstrate that microbial communities can be a selection unit. A protocol for artificial selection of groups has been proposed by Michael Wade. So in this protocol, you set up several populations of beetles, and there is a given number of adults in each population. You let the population grow for a given period of time, and after this period of time, you measure a trait, a property of this group. And for Michael Wade, it was the number of adults in each population. And depending on the treatment, he selected different kind of population. For example, in the high group selection, he selected the groups or the population with the highest number of adults. In the low group selection, he selected the population with the lowest number of adults. And there were other treatments, such as this one, the C treatment, where there is no selection, so it's only natural selection in experimental condition. And the last one, the random selection, is a kind of control where you select randomly the groups among the 48 population. Then, based on the groups that you select, you inoculate new populations with the same number of population with the same number of adults per population, depending on the treatment. And you repeat this procedure a given number of times that you can call a generation of groups. So it's not the generation of the individual. So we transpose this protocol to the selection of microbial communities. It is almost the same, except that you have different species with microbial communities. And you can have small variations in the composition due to an initial sampling error when you pipette your microbial community in your flask or wheels. Then you incubate your microbial communities, and at the end of the incubation time, you've got stronger differences in composition that you had initially because of the specific dynamics of the community in each flask or wheel. Then you measure a trait. It depends on your interest. For example, in the literature, people selected for high pH or high degradation of pollutants, or in our work for low CO2 emission, for example. And so you choose for example, three flasks among those flasks, and you use the community to inoculate a new generation of flask or micro ecosystem. In our work, we did not use flask, but we use microcosm filled with natural soil, and we plant them with a plant, and we selected the microbial communities which had the higher impact or the best impact on the plant phenotype which develop in this soil. So our experiment is about artificial selection of rhizospheric communities associated with change in plant traits, and the trait that we are studying is the greenness. So I will explain you. So for this experiment, we use the plant Brachypodium distachyon and a soil microbial community found in the wild. The selected trait was an index of greenness, which was calculated based on phenotyping images, where we calculate the green intensity and the leaf area, which are related to the nitrogen uptake. What is important is to have non-destructive measurement to avoid disturbing the microbial community. And the experimental design was made of 3 treatments, 1 control with random selection, and 2 selection regime, 1 for high greenness, 1 for low greenness. There were 3 lines per treatment, 20 plants per lines, and we choose 3 among these 3 among these 20 plants at each generation, and we made it for 10 generations. So to be clear, the plant is not evolving in our experiment. The genotype of the plant stays the same from generation to generation because all the seeds are coming from the same seed batch. What is changing and what is transferred from parental generation to offspring generation is the Rhizosphere microbial community only. So these are the results. So is clearly not as in our dreams. There is a huge variation between generations, stochastic variation, which was due to environmental variation. But when you report the values of the greenness index that we select from the high and low line to the control, the random line, then you can see that we can observe for some generation differences between the index between the high and the low line. And if you look in detail, there is a first phase where there is no differences in this initial phase, and a second phase where it seems that the differences are more obvious. But this is an arbitrary distinction between these two phases, and to see if it corresponds to something true for the microbes, we look into the detail of the dynamics of the microbial communities along generations. So on the left, for the bacteria, as you can see from the first to the last generation, we first observe very important oscillation in the structure of the microbial community from generation 1 to generation 5. And then from generation 6 to 10, there were almost no change in microbial communities. And this was not exactly the same for the fungi, but for the fungi, we saw a sharp change between generation 2 and 3, and then a very small change up to generation 10. To be sure that there is really a stabilization in the structure of the microbial community, we look at the data by comparing the structure of the microbial community at the last generation as compared with the structure of the microbial community at the first, second, etc. generation. So the highest the value, the highest the dissimilarity, so the difference between community structure. So as you can see, especially for bacteria, we observe a decrease in the dissimilarity during the initial phase, and then the segmented regression said that there was a breakpoint at generation 5, and after this generation, the structure of the microbial, the bacterial community was the same for the 5 last generation. And the pattern is approximately the same for the fungi, but the stabilization was less obvious. What is important is that we did not decide to put this breakpoint at generation 5. This is the segmented regression that do it by itself. So there is really a distinction from the microbial point of view between the initial and differentiation phase. So when we look at the differentiation phase between generation 6 and 10, we see that there were a positive correlation between the structure of the microbial community in the high line and the greenness index, and the negative correlation of this greenness index with the structure of the community in the low line for the bacteria as well as for the fungi. And then we came back to the plant trait, the greenness, by distinguishing the initial and differentiation phase. And what we observed was that there were no correlation between the greenness of the parent generation, the parental generation and the offspring generation during the initial phase. So there were no significant irritability, but we found a significant irritability for the differentiation phase between the property of the parental and offspring community. So we can conclude that it is possible to select rhizospheric communities for their impact on the plant phenotype. So this is of major importance for agriculture and the Holobionts. The stabilization of the microbial communities can arise after several generations. So what we observe is the way from an unstable point to a stable equilibrium at generation 6 up to 10. Then we observe that when stabilization occurred, there were a modification in plant trait that became heritable. So the criteria of Holobionts to define the selection unit are fulfilled with the selection, artificial selection of microbial community. So to conclude, rhizospheric microbial community can be a selection unit if we can stabilize the structure with a standardized experimental protocol. This open perspective, from a theoretical point of view, it is an empirical evidence in the debate about the nature of selection unit in evolution. And with the house project, we go further by trying to demonstrate that we can select the interaction between the microbial community and the plant. And for sure, there is a very practical interest with this kind of result in agriculture and in medicine. This work has been done by many people, and I particularly want to thank Samuel Jacquiot, who take care about the plant and the microbial community analysis. Thank you for your attention. I can't hear you, Emmanuelle.
Okay, thank you, Manuel, for this presentation and the first result on your project. It's my pleasure now to introduce our next speaker, Claire Rogel-Gaillard. Claire is a research director at INRAE. She's a director of a large research unit laboratory working in animal genetics. She has been a pioneer in integrating microbiome in her research and research question. And she is also in the direction board of a graduate school at Université Paris-Saclay. And she's going to tell you about enteropypig project.
Yes, may I share my screen, please?
Yes, sure. We see your PowerPoint, but it's not.
Okay.
It's not full screen.
Okay. So.
Yes, you have to switch.
Does it work now?
Yes, it's perfect.
Full screen?
Yes.
Thank you.
Switch your camera, maybe?
Yes, I can switch on my camera. Okay, so thank you for the kind introduction. So it's my pleasure to present the enteropypig project that is part of the Holo-Meta program and that was presented before by Emmanuelle. So since Holobionts have been well described before, I can start by the fact that the gut microbiota is a dynamic ecosystem over life. So and you have here this scheme. It is a photo where you have this tight connection between the epithelial, the gut epithelium, and inside in the lumen, the different bacterial community. So when we say that the gut microbiota is a dynamic ecosystem, there are changes during life. And here, for enteropypig project, we focus on early life, between birth and before puberty. So animals are between birth and 120 days of age. And so we are between the colonization of the gut and diversification of the microbiome and the first stabilization and maturation of the gut microbiome. And so we work in pigs and we are interested in looking at the gut microbiota and the links with production traits, with health and welfare traits, and also links with adaptive and resilience traits that are very important for sustainable practice systems. So we have found that there is a stratification of pigs according to two enterotypes, and the enterotypes correspond at different ecological systems. And on 60-day-old large white piglets, so on a group of more than 500 animals, we could identify one enterotype more driven by the two genera Prevotella and Microbiota, and another enterotype more driven by Rhinococcus and Treponema. And here you have this information that these genera, they work in tight connection with many other bacterial genera and species. And here you have these positive and negative correlations with the Prevotella here that work together with the Microbiota, and the Rhinococcus that work together with the Treponema, but they are negatively correlated with the other genera that define the different enterotypes. So this was published in 2016. So our aim was to study whether these enterotypes could be transferred to the next generation in the concept of genetic heritability. So here what we found is that there are relationships between enterotypes and growth rate, meaning that these different ecological systems have impact on the host phenotype. And you can see here that there is a higher body weight gain in 60-day-old animals with fecal microbiota that contain more Prevotella. And you have these two body weight average and average daily gain, and you can see that there is a higher body weight, higher weight rates for Prevotella, Microbiota enterotype. So what are the interactions between the host and the microbiome? And then we go back to the issue of holobiont assembly. And there is a controversial issue on the influence of the host genetics. And you can see here on the different titles of these papers that on the one hand, it is said that the genetics has major impact to shape the gut microbiome. On the other hand, that there is the environment that dominates over host genetics in shaping human gut microbiota. And we have also, and it was very well presented just before me, the way that we need to rethink also the heritability of the microbiome. But I will mostly focus on the heritability and genetics of the gut microbiota from the host part. So the objectives of the enterotype project is first to estimate the heritabilities of the composition of pig gut microbiota at 60 and 120 days old on piglets that are 60 and 120 days of age. A second objective is to demonstrate the possibility of acting on the host genome to modify and to orientate the composition of the gut microbiota. Also to study enterotype functionalities, but we have not already results on this part, and to create pig genetic lines as models to further study host gut microbiota interactions, dynamics of microbiota over life, and links with host phenotypes. So what is our selection criteria? It is a microbiota at 60 days of age. So on this scheme, you have animals, they are born in a farm, they are weaned at 20 days of age. We sample we sample stool when they are 60 days old, and you also sample stool when they are 120 days of age. And for the fecal microbiota characterization, so we produce the data, we extract DNA, and then we sequence the 16S ribosomal RNA gene. Then for this, here you have the sequence data processing with the sequence alignment to the different databases, and the rarefaction set to 10,000 counts, and then we have abundance tables for each operational taxonomic units. And then we have the enterotype determination with the genus relative abundance, and we cluster them based on Jensen-Shannon divergence. And it is according to a paper that was published in Humans, and for which, and we used also the same method to identify the enterotypes in pigs. So we have the selection experiment. So we produce two pig lines referred to as PM for Prevotella and Mitsuchella, and RT for Ruminococcus and Treponema. So we started from 30 families, and we produced in the founders, generation 0, more than 300 piglets, and that were separated according to the 2 enterotypes that we could determine. And we follow, and then we mate again 6 males with 13 females to produce the first generation and to study the selection, if the selection works on this phenotype of microbiota 60 days of age. So here we compare for the generation 1 more 133 piglets to 139 piglets. So in the base population, we have this stratification. We reproduce the stratification of the population in the 2 enterotypes, and one third of the animals with enterotype RT, well balanced for males and females, and 2/3 of animals with enterotype PM. And as you will see, Prevotella is always dominant, whatever the enterotype, but the relative abundances are higher in the Prevotella Mitsuchella enterotype compared to the other one. So here you have the individual variabilities within enterotype groups, but also the differences between the 2 groups. So here each bar corresponds to one individual, and here you have the group of the Prevotella Mitsuchella enterotype, and here the group with the Ruminococcus treponema enterotype. And you can see that Prevotella, whatever the enterotype, is dominant, but more dominant in the PM group. And you have here the 2 examples of the overdominance of Prevotella and Mitsuchella in this enterotype. And for the other enterotype, we have an overdominance of Ruminococcus compared to the other enterotype, and of Treponema also compared to the other enterotype. What we also identified is that even if we have enterotypes at 60 days of age, some of them change according over life. And we have here, if we combine all animals at the 2 days of age, we have almost half of the enterotypes PM that turn to RT, and most of the PM enterotypes remain PM, and maybe half of the RT remain only RT. So it means that even if there is a genetic determinism of being of one of or another enterotype at 60 days of age, there is a capacity of evolution, probably due to environmental and aging. So we performed statistical analysis, and we focused on the 4 genera that define the enterotypes. And we looked at the differences between the 2 pig lines. We used the data from generation 1 just to know if there is a response to the selection. We focused on the 4 genera under selection, and we looked at the pigs at 60 and 120 days of age. And we estimated genetic parameters by using the data from G0 and G1 generation. We use as fixed effects the sex and the batch, and we estimated heritabilities and genetic correlations between data from samples collected at day 60 and day 120. So we can see that there is a good response to selection. Here, if we look at the direct criteria that is a microbiota at day 60, so we have here the question, what is the difference between generation 1, between line PM and line RT? And you can see that there is a good response for the 4 genera with a higher abundance in the PM line compared to the RT line. If we looked at the response to selection at day 120, we have only differences that is partially maintained at 120 days of age for Prevotella only, and that is mostly due to the transfer and to the modification of one enterotype to another one. If we look at the genetic parameters, so for the 4 genera under selection, you can see here in blue, you have the heritabilities of the microbiota 60 days of age. In orange, you have the heritability at 120 days of age. And in gray, you have the genetic correlations between the two parameters. And for the 4 genera, you can see that the heritability is quite high at 60 days of age, ranging from 0.4 to 0.3 to 0.4. There is a higher heritability at 60 days of age, and there are medium genetic correlations, mainly due to the change in the enterotype during aging. So as conclusion and perspectives, you can see that heritability are of moderate values and in same range for the 4 genera, meaning that they are well balanced in the way they shape the enterotype. So the values are between 0.3 and 0.4. So we evidence that fecal microbiota can be modified by directional selection, because if we look at each line, each pig line, the composition of each enterotype is reinforced. And we can see also that enterotype may change with age, meaning that there is a very combination of the influence of genetics to shape a microbiota at 60 days of age, quite early, but there is also an addition of the influence of the environment when animals are aging. So with this project, we can provide good model with these 2 lines to study the respective influences of the host genetics and of the environment to assemble the pig holobiont. So thanks for your attention. I would like to thank for the funding the Animal Genetics Division of INRAE and also the Holoflux Meta program, and for the partnership. So we run this project with Catherine Larzul from Genfis laboratory that is in Toulouse, and all people in my team and the different laboratories that help us also to produce the animals and to analyze the data. Thank you very much for your attention.
Thank you, Claire.
Thank you very much, Claire, for a great presentation. So I'm going to ask the other panelists to come on as well at this point. We'll work through a few of the questions. I think Emmanuelle is coming back on.
Yes, that's it.
Okay, perfect. So great presentations from all of you. We do have a few questions. The first one is for Manuel. How does the selection of the whole microbiome, how is that different from traditional ways to inoculate just one or a few of the strains with defined beneficial impact? Is the effect more stable or enhanced under different environmental conditions, for example, with climate or with soil?
Okay, so if I understand well the question, the main difference between what we have done and what is usually done is that people select strains based on what they do in petri dishes. Not always, but often. They look, for example, for nitrogen fixing bacteria or hormone producing bacteria. And because they think that this mechanism is beneficial to the plant, they inoculate the strain in the field. Here, we do not have any idea about the mechanism. We only select the microbial communities for its impact on plant genotype. And it requires additional research to really identify the mechanism. But in fact, we don't need really to know the mechanism to get a positive effect that we expect. So this is one major difference with what has been done previously in Rhizosphere research to improve plant health or plant nutrition by inoculating strains. For instance, we have no idea of the stability of the communities that we could inoculate in the field. There is no data showing the stability of this community in the field. So on this last point, I cannot answer.
All right, let's give Claire a question with regards to the enterotypes. The scatter plot makes it appear as though there's not so much to enterotypes, as a continuum between two poles. How arbitrary is the cutoff between the two?
Indeed, can you hear me? Yeah. Indeed, it is based on the statistics between the differential abundances between the genera that define the two enterotypes. And so the two, if you look at the centroid of the two groups, they are well defined. And indeed, it is the distance to the centroid that define the two groups. But of course, there are some animals that could be at the continuum. But that is the way we define the two enterotypes, by looking at the statistical different abundances between the genera that define the enterotypes. And it's quite stable, because we can reproduce these two types, these two enterotypes in different populations. And yeah, but what we see is that the enterotypes can move to another one when animals are aging, and that is also what is reported in humans. In humans, they have identified few enterotypes, but they can change over life. And that is important to have in mind, that maybe in animals, when we refer to these enterotypes, what we show is that there is a kind of genetic potential. The genetics provide a potential to have one enterotype or another one quite early, at 60 days of age, but it is not a definitive, maybe it's not a definitive enterotype when animals are aging. But the definition of the enterotypes are really based on these statistical differences in the abundance, in the relative abundances of the main drivers of the enterotypes.
All right, so I'm going to go back to Manuel at this point. Do you know if the microbes evolve in your experiment, or if the effect that you see is just driven by the ecological dynamics, for example, in particular, changes in species abundances? Oh, you're muted, Manuel.
Sorry. Yes, this is a very important question to really consider, that the community can be a selection unit. But in fact, we did not look into the detail if there were some mutation in individual species. And in my mind, this is the only way to look if what we observe is the evolution of individual species, or if it is only due to change in abundance and composition of the microbial communities. But about this point, there are interesting paper of Alexandra Penn, who shows that by modeling, she shows that you could have evolution at the community level without genetic mutation, only by change in the abundance of species or the composition of the community. So there are really an interest to consider the community as a selection unit, because you can have additional leverage for evolution by this modification of individual and interspecies interaction. This is a new degrees of freedom that we could act on to move the properties of a microbial community.
All right, Claire, the next one is for you. How can we differentiate between dynamic and mature, stable microbiota in pigs?
What we find is that there is a first step. We see there is a step of diversification, and we have the weaning age, and then there is still a diversification, and then if we have a follow-up of the microbiota at different ages later on, we can see that there is not so much modification of the microbiota. But we still, as I showed on the first slide, we work on young animals, meaning that there is a first step of stabilization, post-weaning. And this first step of stabilization, we find that it occurs at between 40 and 45 days of age. And then there is a first step of stabilization. But if we compare piglets with their mothers, they have a more mature microbiota, meaning that there are also a more complex diversification that can occur later on. But if we look at the period, the growing period for pigs, we can see that during this growing period there is it's quite stable, and the feed can change. But during the growing period, we can consider that the gut microbiota is quite stable in constant environment.
So, Emmanuelle, before we move to a couple of other scientific questions, I have one for you. Obviously, this is a very broad program. How difficult has it been for you to try to bring together all of the disciplines, both the animal disciplines, the plant disciplines, the microbial disciplines, etc.? And do you have any suggestions for how other people can try to work to get around those difficulties?
Mm-hmm. Thank you for the question. Actually, we were amazed for those meta programs, because we were expecting much difficulties, but in fact it's quite easy, because if you put, scientists, they love to talk to each other. So if you put a bunch of scientists in a room and you ask them to think about priorities, programs, they start discussing about their key question, what they're doing and stuff like that, and it's not that difficult. I have to say that the only difficulty we have right now, but probably also because the meta program is starting, so it's a bit new, is with people from more human science, societal science, which are, it's more difficult because we are really used to the field of experimental science, and they are really in a different field. But even with them, you need time. You need to force a little bit people to work together. But and scientists who the project are working very well for multidisciplinarity and to get those people joining. I think it's also important to realize that the questions which are addressed can be addressed in animals, in plants, in humans. We have the same type of questions, in fact, So it's very, very interesting and very enriching to get those different disciplines and people working together. Yeah.
So just to follow that on a little bit, one of the things that we're very interested in the Phytobiomes Alliance is, are there potential relationships between what we're seeing in the plant, on the plant side, and the feed that then goes to the animal, and what does that impact look like? And are you beginning to think about, from a standpoint of the future, how you pull these two, great projects, how do you pull them together and go to that next level?
Yes, it's a quite important question, and we don't have a program on that already, but it's one question we are discussing a lot. So, well, I don't know if I should say that. I think our scientists at this stage are quite convinced that there is a link, and that we should consider better the practice and production method for the quality of feed and food at the end, and that there is those microbial flux or flows are really impacting not only in one area of the food production system, but to several ones. Yeah.
All right, well, we've got time for a few more questions, more technical in nature. So let's go back to Claire. Can you specify how you calculated heritability?
So we used the relative abundance of each genus, and then we used it as a phenotype.
Okay. Yeah. All right, Manuel, are you aware if there are similar studies that have been performed to stabilize microbiota from the phytosphere?
Microbiome from the?
Microbiota from the phytosphere.
No, I'm not aware of such studies. From what I read, I know only 2 other papers, and both are about the rhizosphere. And in my opinion, there is a very strong connection between soil microbial communities and the plant. And it seems to me that rhizospheric communities have a huge impact. So maybe it's easiest to do it with the rhizosphere community. But I don't have seen any studies about the phytosphere.
All right. So let me just follow that up with another one for you, Manuel. What do you think is the basis for the selection of the whole microbiome, as it's different from traditional ways to inoculate just one or a few strains with defined beneficial impact? Do you think this is the effect of more stable or enhanced under different environmental conditions?
I can't show you this result, but we have done a post-selection experiment in which we used another soil and totally different. We passed from a sandy soil to a clay soil, and we could observe the same effect that we get in the sandy soil. So, but we also change the plant species, but we totally lose the effect. So it seems that the stability of the microbiota is really dependent on the plant species. But with the plant species, you can change the soil and the effect remains. As compared with inoculation of strains, as I answer to the first question, it is really different in my opinion. And so it's difficult for me to compare the two. And once again, I don't know what happens with this community in the field.
So one last question for you, Claire, and kind of getting back to this issue of looking at what's going on with the plant microbiome, and in particular, maybe even what is going on with regards to a seed microbiome that's coming in with the animal feed, or what may be happening in the processing of animal feed for the pigs. Is there any relationship, you think, between that? And has there been some studies where we could begin to start looking at those patterns and how they might fit against the microbiota or the gut microbiome of the pigs?
Indeed, there are many studies aiming at studying the impact of different feed diets on the microbiota composition. What we need to do is to combine the genetics, the feed, and the microbiome response in order to understand how all these components shape the holobiont, I would say, phenotype. Because it would be too bad that on the one hand, the genetic, all people working in genetics improve animals without looking at microbiome, and people try to modify microbiome by feed additives or anything without knowing what the genetic variability can help or can modify. So it would be really too bad that on the one hand, genetics improves farm systems.
Right.
That is not compatible with what feed additives try to do, and there is a kind of compensation that does not work in harmony. So really, for me, the big deal is to both study individual variability at the genetics and microbiota levels together to understand how these two types of variabilities shape the holobiont, and provide good services, good quality products, good impact on the environment. So we cannot work on biodiversity only on one side. We have to work on biodiversity on genetic side of the host and genetic side of the microbiome. And then we can study many different environmental things that could modulate the final response at the holobiont level. So really combining, because you see the people work sometimes separately.
Yes, that's the challenge.
So this type of I think Phytobiomes Alliance, like the Holo-Meta program, can help to build this general view of combining the different biodiversities to shape holobiont and to know how this combined biodiversity respond to various environmental challenges or anything, threat, pathogen threats, immune response, immune capacity, many things. The big issue is to work on the two types of biodiversity that work together.
Exactly. It's those interactions and how they all come to bear.
And if I may add something, you see how similar the questions are with human holobionts and diseases, you know, infectious and chronic diseases. It's exactly the same. We have a diversity of response, which really needs to question the diversity of human genetics and epigenetics, of microbiome diversity and enterotypes, and of food effects on the system, on the holobiont. So it's really, really close.
At least from my perspective, I think in agriculture, we're poised in a much better situation of, you know, grappling with this because we can measure things more easily in our livestock and in our plants. We can do that much more rapidly than they can in the human systems. Mm-hmm. Maybe we will lead the way in this regard. So,
yes, yeah. Maybe. Yeah.
So I want to thank Emmanuelle, Manuel, and Claire again for being with us today. There were some questions that have not been answered, but we will try to answer those, and in fact, perhaps even attach them to the presentations on the YouTube channel. So I'd like to thank you again for being with us, and would encourage you to look into the Meta program and the opportunities for collaborating with INRAE and collaborating, of course, with Phytobiomes Alliance. But thank you for your time today, and we look forward to seeing as many of you as possible at our next webinar.
Thank you, Kelly, and thank you to Phytobiomes Alliance for this opportunity to share our program and our projects. Thanks a lot.